Hi, welcome to part 6, DNA and cell division. Three major or key ideas here. The DNA structure is important to know and understand. A little bit about the cell cycle, why organisms have a cell cycle, what does it do, and then how is mitosis and meiosis different, similar to each other, and those are both types of cell division. So there's lots of vocabulary here, many of the words we've already used, like DNA is an example of a nucleic acid. Nucleotides are the building blocks of these nucleic acids. And I think we've used, and that's it. That's what we used so far. So let's start with the questions. What is the structure and function of DNA? So DNA's structure is a double helix. Okay, so that's that twisted ladder thing, right? Double helix. It's composed of nucleotides. All right, those are the building blocks of DNA. A nucleotide is made out of a sugar bonded to a phosphate, right, and bonded to a nitrogen base. Right, so those are the three parts, and they're bonded together. The sugars and the phosphates make up the backbone or the sides of the DNA molecule, while the bases go across from side to side. All right, so what is the function of DNA? So the function of DNA is to code for proteins. Right? So the order of the bases determines the amino acid. An amino acid plus an amino acid makes a protein. So the function of DNA is to code for proteins. Some people say that it is the information for the cell to function. Right, so you can also say that information is cell to function, and it does that by coding for proteins. That's probably the full definition right there. Okay, so on to the next question. How do base pairing rules ensure accurate DNA replication? So first, let's go over the base pair rule. In DNA, remember we said we have nitrogen bases. The nitrogen bases are the letter A for adenine, T for thymine, C for cytosine, and G for guanine. You just have to use the letters. A always bonds with T and C always bonds with G. And those are the base pair rules. So how do these rules ensure accurate DNA replication? When DNA is ready to replicate, and this happens during interphase, right? During interphase, we have a G1 portion, an S, and a G2 portion of interphase. It is during the S portion, the synthesis portion, that new DNA is made. A cell is going to be going through this cell cycle to make new DNA only when the cell needs it. Okay, so if you have a strand of DNA that has the following sequence of letters on a strand, all right, remember it's double stranded, but in order to replicate it, you have to pull it apart. Then when you go to make your copy, and you follow the base pair rule, T always pairs with A always pairs with T, and you should be able to do this with a strand of DNA provided to you. So I've done that, and it was pretty darn simple to do because C's always go with G's and T's always go with A. There's no other way to do it. So when you go in the synthesis part and make a copy, it's always turning out perfectly because A's go with T's and C's go with G's. There's no other letters involved here. Now, there's also an enzyme that comes through and it double checks to make sure that things are in the right spot. If they're not in the right spot, it goes ahead and corrects it. So all of this is happening during the S portion of interphase. The next part of the cell cycle is something called mitosis. Mitosis is made up of prophase, metaphase, anaphase, and telophase. And you don't have to know specifically what happens in each one, but you need to know that this whole process right here, its job is to divide the nuclear material. Nuclear material is DNA. It's chromosomes. So all of mitosis does that. And then once mitosis is over with and the chromosomes have been pulled apart from each other, the last part happens, and that's cytokinesis. Spelling it wrong. And cytokinesis is the splitting of the cytoplasm. The whole purpose for the cell cycle is for growth. You didn't start out the size you are today. You had to get more cells to get there. Repair. Have you ever broken a bone or scratched your skin? And if you're a bacteria and a few other organisms, you use it for asexual reproduction. That is the purpose to go through the cell cycle. And this is known as, sometimes it's just referred to as mitosis. But the whole thing really is the cell cycle. Let's look at what that would look like as a drawing. Typically, you start out with one cell, and a chromosome number is placed on the inside of that cell. And this is labeled the parent cell. If this cell undergoes mitosis, when it's all done going through interphase, mitosis, cytokinesis, the whole process, it goes through that whole cell cycle, there should be two cells at the end, and they're called daughter cells. And they will have the exact same number of chromosomes as the parent cell because they are identical to the parent cell. Now, this 2N right there that I put in that cell, that represents this vocabulary word right up here, diploid. So if something is diploid, it means it has a full amount of chromosomes, and that is abbreviated as 2N. When cells use the process of the cell cycle for growth, repair, and asexual reproduction, you're getting an exact copy of the parent. There's no variation, no genetic variation, no variety. It's exactly the same. The opposite of that, or a little different from that, is something called meiosis or meiosis. So in meiosis, you start out with your parent cell, which is 2N, which means diploid, means full amount. You're going to go through doubling your DNA, just like you did in our cell cycle over here in the synthesis phase. And then you're going to split the cell twice. So you're going to end up with not 2 cells, but 4 cells. And you're going to end up with half of the original amount of DNA. So these can be referred to as daughter cells. However, a better name for this is a gamete. And this is either an egg or sperm. Each of these are haploid cells, meaning they have half of the parent's DNA. All right, so you have half of the parent's DNA. This is important because when 2 gametes join, when egg plus a sperm join up, we get back to our full amount. All right, so we get back to a full set. So egg is an N, sperm is an N, and then when you put them back together, you get 2N. You get back to your full amount. So a haploid cell and a haploid cell get you back to a diploid cell. Now, this new full set is a unique genetic combination because it's 50% from the mother and 50% from the father. Easy ways to tell them apart in a drawing are the number of daughter cells. There's 2 here, and they're identical to the starting cell. In meiosis, it's 4 cells, and they are half of the original parent cell. So if we were going to do this with a number, if we said the parent cell started out with 14 chromosomes, what would each sperm have? You would have 4 sperm with only 7 chromosomes in them because it's half of the parent's genetic information. All right, so meiosis gives you genetic variation, where mitosis does not. That answers all the questions on the study guide. If you have any questions, please come and see me. Thanks.